Hello, class. Today we'll be looking at the nervous system, specifically nervous tissue. What are the main organs of the nervous system? When we're looking at the nervous system, it contains the brain, spinal cord, nerves, and then the receptors. When we're looking at nerves, they're broken down into cranial nerves and then spinal nerves. What are functions of the nervous system? The overall role of the nervous system is communication, and that communication allows for maintaining homeostasis, which means balance. So, one of the things is sensory input. So, your nervous system is able to detect any type of stimuli that comes in, whether that's external or internal. Okay, external meaning outside in the environment, so things like temperature and touch, or internal dealing with changes in blood pressure or internal body temperature that needs to be regulated. The next thing is integration. So after those signals are detected, the next step is for your brain to process that information and interpret what needs to happen. Another role is the control of muscles and glands. So your nervous system sends signals to the different organs in your body and allow for gaining control. Then homeostasis. So that's maintaining balance and making sure things in your body are at a consistent rate or set point in order for your cells to function properly. The last thing is mental activities. So mental activities can involve memory or seeing or just interpretation of any type of signals. So let's look at parts of the nervous system. So when we're looking at the nervous system, it can be broken down structurally and then functionally. So first we're going to look at the structural breakdown of the nervous system. So the nervous system can be broken down into the central nervous system and then the peripheral nervous system. The central nervous system contains the brain and then the spinal cord. When we're looking at the peripheral nervous system, it contains all of the nervous tissue outside of the central nervous system. So this involves the sensory receptors and then the nerves. After the structural classifications of the nervous system, it can also be broken down functionally. So the first functional category is the sensory division. So the sensory division deals with incoming information. So any type of external or internal stimuli is sent towards the brain in order for interpretation to happen. Then you have the motor division. So this involves output. So once information is sent to the brain, for it to be interpreted, then the brain can send out signals in order for your organs to carry out any necessary activities, to carry out an action you want to do, whether that's walking or to maintain balance. The motor division can then be further broken down into subcategories. So you have the somatic nervous system and then the autonomic nervous system. The somatic nervous system is also known as the voluntary nervous system. So this involves skeletal muscle. So in the previous lecture, we talked about how skeletal muscle deals with voluntary control. So you control how your muscles work. So this is the somatic nervous system. So your brain sends signals to the muscles that you want to move. Then you have the autonomic nervous system, which is also known as the involuntary nervous system. So this involves cardiac muscle, smooth muscle, and glands. So these particular structures you do not consciously control. Okay, your brain automatically controls these organs in order to maintain balance or carry out necessary life functions. The autonomic nervous system can then be broken down into further categories. The two most important involve the sympathetic division and then the parasympathetic division. So the sympathetic division deals with fight or flight. So this is where your brain is sending signals when you're doing a lot of physical activities or you're scared. And then when we're looking at the parasympathetic division, this deals with resting functions, so rest and digest. When your body isn't in high alert or is undergoing a lot of activity, then other functions begin to become more active. Okay, where sympathetic division would be you working out, so a lot of signals would be sent to your skeletal muscles, cardiac muscles for your heart to beat and pump blood through your body. Then when we're looking at the parasympathetic division, this would be when you're resting. So a lot of other functions take over during that time, such as digesting foods or removing waste from the body. The last division of the autonomic nervous system is the enteric nervous system. So the enteric nervous system deals with the digestive system, and it is the main nervous system that controls your digestive system. Your parasympathetic division helps to regulate the digestive system, but the enteric nervous system has the main control over how foods move through your body and what's digested and the enzymes that are released. So here's just looking at an example of how these two nervous systems work together. Okay, your peripheral nervous system and then your central nervous system. So let's say you go out camping and you encounter a bear. So that is the stimulus, right? You're going to run. So if you see a bear out there in the wild, you have your sensory receptors. Your eyes are going to see the bear, and that's going to send signals to your brain in order for you to respond to what you're seeing. So we're going to say fight or flight, right? So we're going to send information to the motor division to begin to tell your skeletal muscles, hey, it's time to run. And then also send signals to things like cardiac muscle that will cause for your heart rate to increase, which will allow for more blood to travel through your body. Your smooth muscle might contract. That can involve your blood vessels beginning to constrict in order to increase the flow of blood. And then glands. So you may start sweating as well out of fright or because you're hot because you're running so your body can cool you down. What are the two main cell types of the nervous system? So when we're looking at the nervous system, you have neurons, and then you have non-neural cells, which are also known as glial cells. You might hear them called neuroglial cells. So the neurons are the ones that actually send signals. Okay, so they're the conducting cells. Then you have the glial cells, and these cells help to support the neuron. And later on, we'll talk about each of these different types of neuroglial cells and what their role is. So first, looking at the neurons. So neurons have three main parts. You have the soma, which is the overall cell body. So remember, in the cell body, you have the nucleus, nucleolus, other organelles that are in this area. You have other extensions off the cell body, like the axons and then the dendrites. So the dendrites are the receiving extensions of the neuron. Incoming information moves through the dendrites to the cell body where information is processed. And then outgoing information moves through the axon, which will allow for the neuron to communicate with other cells or send signals. So here's looking at a neuron. So let's look closer at this image and talk about some of the different parts. So this is looking at a neuron connecting to another neuron. So we already talked about the dendrites that are coming in. This allows for signals to come into the neuron in order to be processed. You have the entire cell body here, okay, which is also known as the soma. So inside of the soma, you have the nucleus, What's the role of the nucleus? So the role of the nucleus is that it houses DNA, okay, which is the blueprint for your cells. Inside the nucleus, you have the nucleolus. Do you remember what the role of the nucleolus is? So the role of the nucleolus is ribosome assembly. Okay, remember the role of ribosomes is protein synthesis. Moving to other structures, okay, we talked about the axon. So the axon allows for neurons to send out information. Where the axon originates is known as the axon hillock. And a lot of the signals that are coming into the neuron begin to build up. And there has to be strong enough signals coming in order for this neuron to actually send information out. At the ends of the neurons, you have synaptic knobs, and this allows for neurons to communicate with other neurons. Although you do not see them labeled in this image, neurons also have mitochondria. Remember, the role of mitochondria is that it deals with ATP, powerhouse, energy for the cells. You also have rough-ER and ribosomes, but those are known as nestle substances, okay? Nestle substances or nestle bodies. And that's just a combination of ribosomes and rough-ER. And remember, the role of those organelles are protein synthesis. One thing that organelles lack are centrioles. And if you remember centrioles, the role of centrioles is cell division. And because neurons lack those particular organelles, they're not able to undergo cell division, which means they cannot replicate. When you damage these cells, they do not regenerate. So we just went over some of the different parts of the neuron. So here's looking at a micrograph of a neuron. So let's go through some of these parts and see if you can identify them. What is this image highlighting? So this is looking at the axon hillock. Okay, remember the axon hillock is where the axon originates. What is this image highlighting? So this is highlighting the soma or the cell body, okay, where the neuron processes information. Next, what is this highlighting? So this is highlighting the neuron, okay, which houses DNA. What is this highlighting? This is highlighting the axon, where you have the output of information. What is this image highlighting? This is highlighting the dendrites, where you have the input of information. And what is this highlighting? This is the nucleolus, okay, and this is the site where ribosomes are assembled. And then what is this highlighting? So this is highlighting the nestle substances or nestle bodies that I spoke about earlier. Okay, and remember, these nestle bodies or nestle substances are the combination of rough-ER and ribosomes within the neuron, okay, and the role is protein synthesis. And if you can see, there are tons of these structures in a neuron. Neurons tend to have more ribosomes and rough-ER than other cells because they need to produce proteins, specifically channels that allow for communication. What are the functional classifications of neurons? So neurons can be broken down into functional and structural classifications. So we're first going to look at the functional classifications. So the different functional classifications of neurons are sensory neurons, motor neurons, and then interneurons. Sensory neurons allow for sensing. Okay, so that deals with the input of information to be sent to the central nervous system. So, detecting external stimuli, internal stimuli, that information is going to move through a sensory neuron. When we're looking at a motor neuron, that deals with output. So, your central nervous system communicating with organs in your bodies or effectors, that's going to deal with motor neurons. Then you have interneurons, also known as association neurons. And this allows for information to be coordinated between incoming and then outgoing information. Here's a video that will walk you through what happens in your nervous system when you interact with some type of external stimuli. And also look at the different functional classifications of neurons and just give an example of their functions.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body.
What are the structural classifications of neurons? So, when we're looking at the structural classifications of neurons, it deals with how many extensions come off of the cell body. So, let's take a closer look. So, when we're looking at multipolar neurons have several extensions that come off the cell body. Okay, you can see all of the dendrites that are extending off, and then you also have the axon. So, with multipolar neurons, they tend to involve a lot of incoming information and then also outgoing information as well. When we're looking at bipolar neurons have two extensions that come off the cell body. So, you have a dendrite and then an axon. Then looking at unipolar neurons. So unipolar neurons only have one extension that comes off the cell body. So you have the cell body here, and all you have is this one process that's coming off. When we're looking at the different roles of these structural classifications, motor neurons and interneurons tend to be multipolar neurons. They're dealing with a lot of incoming signals from the brain that needs to be sent out towards effector organs. When we're looking at bipolar neurons and unipolar neurons, these two types deal with sensory information. So if you look at bipolar neurons, information comes in and moves in one direction. The thing about bipolar neurons, you tend to find them more in children, and then they deal with sensory information like sight and smell. When we're looking at unipolar neuron, the same thing, okay? They bring in sensory information and kind of move in a one-way direction. And this allows for other types of sensory information around your body. You can view this chart in order to break down and differentiate the different roles of these structural classifications. What are the types of glial cells? So we talked about how glial cells help to support the neuron. So here you can see the two different neurons and then all of the glial cells that surround those neurons. So let's take a closer look at these neuroglial cells and their functions. So first, looking at the astrocyte. So if you can see these green cells, they help to anchor neurons to blood vessels. This allows for astrocytes to send nutrients to neurons. Neurons have a very strict ion composition that's needed in order to allow for communication and send signals. Your blood varies based on what you ingest and just your different body needs. And the neuron cannot deal with the fluctuations that occur in blood vessels. So the astrocytes help with filtering needed nutrients for the neuron. Next, moving to the ependymal cells. So the role of the ependymal cells is that they can produce cerebral spinal fluid, and cerebral spinal fluid surrounds nervous tissue, and it helps to maintain the chemical needs and chemical composition of these nerve cells. They also contain cilia on their surfaces, which are going to allow for the overall movement of cerebral spinal fluid through different cavities. Next, looking at the microglial cells. So the role of the microglial cells is they help with immunity. Microglial cells are phagocytic cells that can ingest any type of harmful bacteria or viruses that may damage nervous tissue. They also have processes that can monitor the health of neurons. Next is looking at the oligodendrocytes. So the role of the oligodendrocytes is that they help to myelinate the axons of certain types of neurons. So myelination, which we'll go further into later, helps to protect the neuron, but then also allows for the neuron to send signals a lot faster. These different types of neuroglial cells are found in the central nervous system. So astrocytes, ependymal cells, microglial cells, oligodendrocytes, these are found in the brain and then the spinal cord. Now looking at some of the different neuroglial cells that are found in the peripheral nervous system. So here's looking at the satellite cells. So the satellite cells help to insulate the cell body, which also allows for the regulation of nutrients and then waste. So satellite cells aid in protection. Then you have the Schwann cells. The Schwann cells are what myelinate neurons in the peripheral nervous system. Earlier, we looked at the oligodendrocytes that help to myelinate neurons in the central nervous system. So the Schwann cells do this, but in the peripheral nervous system. And anytime we're talking about myelination, it helps with allowing for neurons to send signals faster. What is the function of myelination? Do you remember what I just said? So it helps for neurons to send signals faster. So remember when we're looking at myelination, you have Schwann cells and then the oligodendrocytes. Schwann cells wrap neurons in the peripheral nervous system. Oligodendrocytes help to wrap cells in the central nervous system. Okay, and both of these form myelin sheaths. And myelin is this lipid-based coating that helps to protect the neuron. It also helps to insulate, and insulate allows for ions to be closer to the plasma membrane so electrical signals can be sent faster. So myelinated neurons send signals faster than unmyelinated neurons. So here's looking at the difference between myelination in the peripheral nervous system with the Schwann cells. You can see that each Schwann cell, which are individual cells, wrap certain areas along the axon. Then we're looking at the central nervous system and the oligodendrocytes. They surround parts of the neurons within the brain spinal cord. So when we're looking at the central nervous system, there are certain terms that we need to know. White matter versus gray matter. And then when we're looking at white matter, the difference between nerve tracts and nerves, and then the difference between nuclei and ganglia. So when we're looking at white matter versus gray matter, white matter deals with myelinated neurons. Okay, remember myelination deals with this lipid-based covering, which tends to be white in appearance. Then you have the gray matter, and that deals with unmyelinated axons along with the cell bodies. Okay, remember the soma. Going back to white matter, when we're looking at axons, okay, nerve fibers, and remember axons are what send out information. When we're looking at groups of nerve fibers or axons in the central nervous system, they're known as tracts. When we're looking at groups of axons or nerve fibers in the peripheral nervous system, they're known as nerves. Okay, so they're both the same structure, but we call them different things depending on which structural classification we're looking at. So they're tracts when you're looking at them in the brain. They're called nerves when you're looking at them in the peripheral nervous system. Then you have gray matter. So remember, with gray matter, unmyelinated axon cell bodies. So when we're looking at nuclei, nuclei involve groups of cell bodies in the central nervous system. When we're looking at groups of cell bodies in the peripheral nervous system, this is known as ganglia. A synapse can be formed between neurons, and synapses are cell-to-cell connections. So you have the end of one axon at the axon terminal connecting to maybe a dendrite or the cell body of another neuron. So here's looking at the location of gray matter and white matter in the central nervous system. So first looking at the spinal cord. So with the spinal cord, gray matter is found internally, and then white matter is found on the outer surfaces. So the white matter allows for sending and receiving information from the brain. The gray matter allows for the information that is being sent to be processed. So you have myelinated axons in the white matter, and then you have unmyelinated axons and cell bodies in the gray matter. Then looking at the brain. So when we're looking at the brain, you find gray matter in the outer cortex, which just deals with the outer edges of the brain. And then you find white matter in the internal parts. So the internal surface of the brain deals with a lot of myelinated tracts, which are sending information to the cortex. And the cortex, or gray matter, is where all the information is processed, whether that's dealing with temperature or movement or memory. And then any outgoing information or incoming information then moves through this white matter. Describe resting membrane potential. This video is going to review resting membrane potential.
The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization.
Describe the mechanisms that maintain the concentration gradient. So when we're looking at how the plasma membrane maintains its resting membrane potential, remember you have sodium on the outside of the cell, potassium on the inside of the cell. There have to be ways to regulate that, especially since you have so much fluctuations in internal and then external fluid. So mainly you have the sodium potassium pump, which moves ions by active transport. And remember, active transport deals with ATP using energy in order to move ions across the plasma membrane. The sodium potassium pump helps to move potassium into the cell and then sodium outside of the cell. Other ways that the concentration gradient is maintained is you have leak channels. So you have both potassium ion channels and sodium ion channels that are always open. And this allows for maintaining resting membrane potential, where if it becomes too negative inside of the cell, then you can have positive ions move inside. If it becomes too positive in the cell, then those ions will leave the cell. You also have other gated ion channels like ligand gated ion channels. And remember, with ligand gated ion channels, you need a chemical to bind to the channel in order for it to open. You have voltage gated channels, which deal with changes in the voltage or electricity of the plasma membrane that cause for these channels to open. And then you have other gated ion channels, which can deal with touch or temperature, that also help for ions to move. So here's looking at the leak channels. So leak channels are always open. So here, these are potassium. So potassium ions can enter or exit the cell freely. Then you have things like ligand-gated channels where chemicals must bind to these channels in order for them to open. And then you have voltage-gated channels where there has to be some change in the electricity of the plasma membrane that allow for the channel to open. So this is looking at the overall neuron and where you can find certain gated channels. Let's take a closer look. When information is coming in, you have depolarization. So along the entire cell, you have the sodium potassium pump, you have sodium leak channels, potassium leak channels, which are going to allow for maintaining balance. But as you move along the neuron, you begin to find other types of channels like voltage gated channels for potassium and sodium along the way in order for signals to be sent properly. Here's a video to show how the sodium potassium pump works. Remember that the role of the sodium potassium pump is to pump out sodium ions and then to bring in potassium ions. And because this is dealing with active transport, it needs ATP for this channel to become activated.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Here's a video that shows how leak channels work. So remember, you have both sodium and potassium leak channels. And this particular video shows potassium leak channels and how potassium enters or exits the cell based on the charge of the membrane.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from -40 to -90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
This animation is going to show how all of these channels work together in order to send a signal and allow for the cell to go back to rest after the signal is sent.
A neuron is an individual nerve cell that is capable of conducting a nerve impulse. Multipolar neurons, the most common type of neuron, have hundreds of dendrites that receive incoming signals that are transferred toward the cell body. A single axon generates and transmits the nerve impulse or action potential. The resting membrane potential of a neuron exists across the plasma membrane and reflects the ionic differences between the cytoplasmic and extracellular sides of the membrane. Leakage channels, non-gated ion channels, and sodium-potassium pumps help to maintain this polarized state. Sodium is the dominant cation of the extracellular fluid, while potassium is the dominant cation of the intracellular fluid. The presence of the leakage channels in the membrane allows these ions to diffuse across the plasma membrane in direct opposition to the movement of ions through the sodium-potassium pump.